very different to the last talk. I'm going to apologize by starting and saying, unlike Michael's talk, probably not going to be a lot of human data, but hopefully we can agree. If you can see my mouse, just let me know, or my pointer. Hopefully we can agree that there'll be something of value that comes out of mice as far as helping us to understand the complexity of humans as well. So like pretty much everyone here at one level or another, our team is very interested in understanding the mechanisms that regulate the attributes of muscle. And for me, pretty much every slide has to be a simplification. Otherwise, I can't get my head around it. So in many respects, we try to bring things down to simple approaches that we can, I guess, try to deconvolute even further. Healthy people clearly are in a great position as far as being able to maintain adequate protein synthesis relative to degradation. And oftentimes, that is not the case for people in a far worse position, whether it's to do with a heritable condition or an acquired condition or something related to aging, let's say. And you're well aware of the many pathways that could contribute to this shift towards unchecked catabolism. And one of the areas where we have spent a lot of time looking has been around the TGFβ pathway. So again, there are people here who could give a very articulate summary of this pathway and why it's interesting in muscle. What I'd like to do is spend a little bit of time talking about how we're trying to understand the way the TGFβ pathway regulates muscle and where we think we can learn more about particular, let's say, genes that are regulated by this pathway to be able to achieve these changes in muscle mass, whether for good or for bad. So in brief today, what we'll do is we'll summarize by saying that the 30-odd ligands of the TGFβ pathway work in nuanced and selective ways in different cells, but in muscle in particular, ligands such as GDF8 or myostatin and activins will stimulate via their chosen receptors to drive SMAD2/3 signaling, and we think of that traditionally as a negative regulator of muscle mass. Driven to excess, it'll cause catabolism and breakdown. And what I'd like you to try and embrace at the same time is that within the TGFβ network, there are bone morphogenic proteins that operate in parallel, but in an opposite fashion, such that they are driving through SMAD158 to exert a positive influence on muscle, whether that is anti-catabolic or pro-anabolic. And follistatin is nature's favorite inhibitor, if you like, of myostatin and activins. And so it blunts this particular part of the axis, but it also augments and stimulates the positive effects that we see through the SMAD158 axis. And so that's, I guess, the premise for where we have been exploring more to try and understand when you express follistatin in the muscle and you get very profound muscle hypertrophy, how on earth is it doing that? So we've been spending a fair bit of time now looking at this in a range of different guises. And because this particular thematic program incorporates a degree of work around aging, let's just say that one of the things that we've observed is that in older animals that exhibit aspects of sarcopenia, if you give a gene delivery designed to express follistatin with the intent of stimulating hypertrophy, you don't get the same sort of increases in muscle mass that you would see in younger animals. And so you can see here 24-month-old animals achieve roughly half of the hypertrophic effect of younger animals. And if you push out to even older animals, the effect is blunted further still. And that's not because you're not making enough follistatin, and that's not to do with not being able to modulate the SMAD signaling parts of the network. It's something more than that. So we've been trying to understand how that might be, and we've tried to approach that from a range of different perspectives. One of the approaches has been to try and combine our ability with viral vectors to deliver things to muscle and then lay that on top of different types of omic approaches. So this is a fairly busy slide, but what it summarizes for you is that we used a gene delivery-based approach to then capture muscles that were very early in the hypertrophic response when you put follistatin into a muscle, 2 days after the injection when you can't even see an increase in mass yet. And then we also compared those to muscles from animals that had been taken a fair way further out when you've already seen something that amounts to practically a doubling of muscle mass. Processing these muscles for transcriptome and proteome, we were then able to start looking for interactions and relationships there. So on the x-axis here, you see proteomic changes, all in the same direction, whether they're going up or down, versus transcriptomic changes. And the main thing to take out of this is that it's pretty complicated as far as being able to say that there isn't a tight correlation between changes in transcriptome and proteome. Oftentimes everything's going up or down and not necessarily being reflected at the total protein level as well. Nevertheless, within that, we were able to identify some interesting candidates that were going up both at early time points and at late time points, or going down at late and early and so on, and at the level of transcript and proteome as well. And one that caught our eye in particular was ASB2. So this particular protein, ASB2, turns out to be an E3 ubiquitin ligase. And what we were observing was that in muscles that are undergoing hypertrophy because of follistatin expression, you see a reduced transcription, but you also see a reduced accumulation of the ASB2 protein. So you can see here changes in the quantity of protein in these muscles that are expressing follistatin and are growing. We further examined how it might be regulated in the cell by being able to manipulate beneath the level of the receptor. In this particular case, we were playing with SMAD7, which is an inhibitor of both sides of the pathway. And we were also playing with some constitutively active receptors, which I haven't put the data up here for you, so I apologize for that. But the main take home from this particular slide is to show you that if you were to manipulate the signaling beneath the level of the receptors by blocking the SMAD2/3 activity and the SMAD1/5/8 activity, we still find a certain degree of hypertrophy. To simplify again, we generally say that about half of the hypertrophic effect appears through this side of the pathway being blocked and half by this side being augmented. So we don't get as much hypertrophy, but we are seeing that there is still hypertrophy taking place. And in these muscles, we do see a reduction in ASB2 protein, which is quantified here from the blots above. And this is concomitant with the blunting of SMAD signaling on this side of the pathway. So then we decided if it's going down in these models of hypertrophy, what might happen if we were to manipulate ASB2 itself. So we made a viral vector which was able to overexpress ASB2, and we delivered this to the muscles of mice. And we're able to show that by increasing the expression of ASB2 over a period of time, you can see quite significant loss of muscle mass in an otherwise healthy leg. The effects are not dependent on sex. It happens in both male and female mice. It is associated with not only a reduction in absolute force producing capacity, but also the force per cross-sectional area of the muscle is impaired as well, suggesting that there's more to it than just literally a reduction in muscle fiber size. When we tried the opposite approach of trying to knock down ASB2 in a similar manner to what you might see when we're trying to stimulate hypertrophy of the muscle, we are able to affect a mild increase in muscle mass. I wouldn't go so far as to really call it hypertrophy. I think that's maybe pushing the friendship calling it hypertrophy, but it's an increase in muscle mass nonetheless. And it is associated with this knockdown of ASB2 protein. So it does seem that you can take this protein in both directions and have an effect on muscle phenotype, which is interesting to us. So that being the case then, you'll recall we were saying that follistatin put in an old muscle does not cause as much hypertrophy as it ought to, as we would like it to. And we saw something curious when we examined those muscles, namely that there was a reduction in the transcription of ASB2, but compared with healthy animals where you see this reduction in the quantity of ASB2 protein in a muscle undergoing hypertrophy, these older muscles that we're trying to treat do not see the same reductions in ASB2 protein. So then what we hypothesized is that if we were to try and clamp the amount of ASB2 in the muscle, keep it high in a healthy young adult animal, maybe that would reduce the amount of hypertrophy that you could get. And these data on the right essentially show you that. What we found was that if we were to administer a vector that expresses follistatin, we get a certain amount of hypertrophy, but we don't get as much hypertrophy if we express this ASB2 protein at high quantities. So we prevent ASB2 levels from being repressed, and consequently, you don't get as much hypertrophy. That's what this graph is showing. And then here, what we're trying to explain is to say the effect of ASB2 in a healthy leg on its own is to cause a certain reduction in muscle mass. But when you put this protein in a muscle that is trying to grow, the blunting effect on what would otherwise be taking place is even more profound. So what we're trying to demonstrate here is that this is not something that's happening merely in parallel. It's actually something that is contributing and is involved in the process of this mode of hypertrophy. Okay, so what is ASB2 doing? As I mentioned, it is an E3 ubiquitin ligase. It happens to partner with a number of proteins, specifically culin-5 and friends, to be able to form a complex which will then target substrate proteins for ubiquitination. And those of you who are, of course, much better read in the ubiquitination space will understand and appreciate that it's these E3 ligases which confer specificity of the ubiquitin proteasome-based system as well. The E3s specifically ubiquitinate individual substrates and mark them for what happens next. The question of what happens next is, I guess it's variable and it's diverse. We often think of E3 ligases as marking proteins for degradation. Usually that involves polyubiquitination. But monoubiquitination can also alter a protein's function without necessarily changing its abundance, and that's important. We'll get to that a bit more shortly. So there have been some pieces of data published, nice pieces of work that have shown that ASB2 as an E3 ligase can target certain proteins in striated muscle. It has been suggested that filaments may be one set of proteins that are targeted, and this might have some bearing on muscle differentiation. But it may also be that there is a certain degree of targeting of desmin, and that this is particularly relevant for cardiomyopathic states, such that if you overexpress, you can reduce the quantity of desmin, and if you knock down, you get essentially an increase or a retention of desmin in these cells. But we were keen to see whether maybe we could contribute or learn something more there. So the approach that we've taken more recently was to develop some viral vectors which express ASB2 with a tag and administer these to the muscles of animals so that we could study in vivo. And taking samples of tissue from these animals, we then process them for either total proteome or ubiquitinated proteome via unbiased mass spec. And so this graphic or abstract here really just summarizes that what we observed was complicated. There is no simple, easy take home. I guess what I can summarize from here is to say we see marked changes in the total abundance of individual proteins. We also see marked changes in the ubiquitination of different proteins. And this particular graph down here is where we've tried to plot out changes in the abundance of proteins versus totals here versus ubiquitinated proteins here. And so sometimes you see proteins where there is a change in abundance which correlates with its ubiquitination status, and other times there's no bearing whatsoever. So I think what it means is that we've got a lot more still to learn. To give you some very quick snapshots here, fortunately this is all being recorded so that you can go back and look at this in your own time as well. Amongst the proteins that are upregulated in abundance as a consequence of ASB2 expression, we see a number of E3 and E1 and E2 related proteins. We also see a number of proteins associated with the proteasome, but we also see some deubiquitinases as well, which is quite interesting. We also see a number of cytoskeletal and filamentous proteins, including proteins of the sarcomere. In terms of proteins that are downregulated as a consequence of ASB2 overexpression, chiefly we're talking about mechanisms associated with metabolism regulation, a lot of proteins in relation to the electron transport chain. So clearly there are some things happening in relation to mitochondria. And also, again, some changes in the abundance of proteins related to the sarcomeric structure and the regulation of excitation, contraction, coupling. Again, I apologize for going through this so quickly here. I'll just show you a quick snapshot of some really, what I think are fascinating examples of where there is a relationship between ubiquitination and the abundance of the protein itself. So that here we've just got 6 examples, if you like, 3 different myosin heavy chain maps, essentially showing you here that there can be individual sites that are altered in their, either increased or reduced in their ubiquitination, and yet not necessarily changes in the abundance of the proteins. So you can see here a marked number of increased sites of ubiquitination, but not necessarily a change in the total amount of that protein. And those of you who like to skip ahead have probably noticed this one already here. Titin, to me, really, obviously a very large protein, but even for its size, remarkable that it has so many increased ubiquitination sites as a consequence of overexpression of ASB2, and yet no change in total protein in this particular instance here. So suffice to say, we're pretty keen. Thank you. We're pretty keen to understand exactly what that might mean. We've also gone and tried to understand a little bit more as far as what might be the specific targets of ASB2, because I think it should go without saying practically that some of these changes in ubiquitination, some of these changes in protein abundance will not necessarily be directly the effect of ASB2 acting on the substrate, but it may be a secondary effect of ASB2 acting on something else upstream. So in an effort to try to get to that problem as well, we have developed some approaches where we've made viral vectors and constructs that express modified forms of ASB2. In this particular case, a version that has been deleted for the SOX-box domain, where it would normally form interactions with cullen-5 and some of the other proteins to be able to make this complex for loading substrates. And so we've tried to use this approach of a comparison between the wild type version and the mutant version to see whether we can try and understand which particular proteins are legitimate targets of ASB2 in terms of their interaction, and others are simply being affected as a consequence of being loaded into the complex when fully assembled. And so again, a lot of work to sort of rush through very quickly here, but these are just two examples to show you differential pull down of proteins interacting with these two versions of ASB2. It's nice to see members of the complex being pulled down and enriched when you use the wild type version. By comparison, they're not pulled down in this mutant version. And through this approach, we've been able to try and identify some specific candidates that might be directly targeted. And we're very keen to see whether they might be able to shed insight into novel mechanisms by which ASB2 might be working. So as a very quick summary then, hopefully this is a whirlwind tour, but what it shows you is that we're very interested in this notion that ASB2 is a negative regulator of skeletal muscle mass, such that if you overexpress it, even in a healthy muscle, otherwise healthy muscle, that's sufficient to induce muscle atrophy. And that's somewhat different from some of the other more commonly studied E3 ligases, where overexpression on itself is not often enough to be able to perpetuate some sort of change in the phenotype. Conversely, knocking it down can also create a certain increase in muscle mass. As I showed you very quickly there, we've observed a number of different changes in terms of abundance, such that we have particular classes and even targets within those classes that we're keen to pursue in terms of both upregulation and downregulation. And there's no clear relationship between the ubiquitination of the protein and its abundance. And I think that that tells us that we're really just on the first step of understanding more. So this was going to be my last slide because I wasn't sure how long we would have. I'll go through this very quickly because I would like to show you 3 more slides at the back, and I think we'll finish on time still. So I'd just like to say I am immensely grateful to so many people for helping us be able to generate this data and continuing to support us. These 3 slides are just a literature walk, just to say why we think this could be really interesting is because if you look in models of growth, you do see that this particular protein goes down in transcription, whether it's overload ablation or in salmon that have been fed to promote extreme growth. When you feed them or overfeed them, ASB2 levels go down. There's also some potential that this might be a significant E3 ligase in the context of physiological rhythm. On the left here, what you see are changes in the expression in the muscles of squirrels over the annual season. And it's suggested potentially that this variation could be involved in energy mobilization when they enter into torpor. I think this particular slide, I was hoping this would be the one that would prompt some talk from the Italians on the call, as I know there are some outstanding circadian rhythm biologists over there. It seems that ASB2 is one of the most extreme in terms of its excursion during the circadian clock. And so I think it would be potentially very exciting to understand the significance of this here. And finally, disease. So we have seen that there is an increase in the expression of ASB2 in muscles after you cut the nerve, so neurogenic atrophy. And again, colleagues of yours in Italy have also made a link between ASB2 expression going up in certain settings of myotonic dystrophy. So I think this is potentially very exciting to try and understand more behind the mechanism of that particular condition. So we started out with the brief of talking about it in the context of aging, but hopefully what I've done is just tried to bring a little bit more relevance into it to say, not only here at the end, but also perhaps all through life, we might learn more by looking at ASB2 in more detail. So thank you for your patience. And with that, I'd welcome any questions.
Thank you, Paul, for a terrific talk. We're 10 minutes behind. Who's got a question?
Paul, do you see, so one of my questions was going to be whether you see an increase in ASB2 in different pathophysiological conditions, and you showed at the end in response to denervation. Is it increased in other catabolic conditions, or is it a neurogenic ligase?
Well, thank you, Andy. So the short answer to that is I think it is changed in a number of different conditions. I was having a look even just earlier this afternoon in some of the very nice data sets that some of your colleagues have published in Cancer Cachexia, and there is some evidence in some of those animal models that ASB2 is altered there, differentially expressed. As you know, we've got to walk through understanding which models are more relevant than others and so on, but I think that there's something to look at further there for sure.